can't do that at the moment, but we can continue to make progress. So I'll just come to a patient. Patient's all about what I treat, what we do. And there's a question at the end of this. So this is a real patient, someone I saw recently. He's about 70, sort of average age. He was diagnosed 5 years ago, asymptomatic disease, managed with active surveillance. He lived abroad, he was teaching in China. He then came back to the UK. He's originally from here because he'd got a lymphocyte doubling time was going up, relatively widespread nodal enlargement. This was his biggest lymph node in his abdomen, up to about 6.5 centimetres. He was more tired than he had been before, and it was considered that he required treatment. He did have some past medical history. He wasn't unwell. His performance status was 0, but he had a long-standing cardiomyopathy. He was diabetic, and he had borderline kidney function. So what to do based on the fact that we've got all these new treatments available? We'll come back to this at the end. It may be it's just an obvious thing. We'll just give ibrutinib because ibrutinib is best, or is it? So I guess that we know that the data for ibrutinib, and this is just one of the trials recently published, the Alliance study, patients over 65, decent performance status with creatinine clearance greater than 40 mils per minute. A comparison of ibrutinib rituximab, bendamustine rituximab with a PFS endpoint. What it showed, that's what it showed. So this was published last year. You'll have seen this, or you may have seen this. So a survival curve for those who aren't familiar with these, looking at 100% of the patients start at the beginning. Everyone is in remission and alive at that point. As time goes on, with each remission or each patient death, the curves drop. And what the study showed is on the lower curve here, there was a greater rate of either progression or death on bendamustine rituximab, the standard arm, compared to two ibrutinib-based combination, ibrutinib on its own or ibrutinib with rituximab. So it shows two things. Rituximab doesn't add anything to ibrutinib, according to this trial, and ibrutinib is better as marked by progression-free survival, so time to progression compared to standard immunochemotherapy. So we should just give him ibrutinib. Or should we? Well, I guess that's the big question. Have we seen the end of FCR or immunochemotherapy? Well, if the publications might have you believe that that's what we should be doing. We should be just throwing out in the past. But I think it's important, and I think I get this from a sort of patient perspective. Taking ibrutinib indefinitely has attractions, I am told initially. Taking ibrutinib forever is less attractive because of compliance. Taking FCR and starting off with chemotherapy for 6 months is not very attractive. It does have side effects, but it's time-limited treatment. In a proportion of people with CLL, it's extremely effective treatment. So 3 studies have looked at this, and this is all retrospective analysis. The largest was the German CLL8 trial, which was FC versus FCR, and this looked at who does well with FCR, a retrospective analysis from the Andy Anderson, and also data from an Italian retrospective. All of them defined good risk by virtue of having mutated immunoglobulin heavy chains. And in this group, and these weren't defined prospectively, there are very long survivals that can be seen, particularly from the US data. At 13 years, more than half the patients still in remission. So 6 months of immunochemotherapy for a 13-year remission, actually not a bad option, providing you're fit enough to have it. We've already heard about costs. In the UK, the reason I don't give ibrutinib is it's not reimbursed in the frontline setting. And the cost effectiveness, this has not been demonstrated. This is just a publication from a couple of years ago. This is looking at the costs of treating CLL by year. And you can see a huge increase. So, I mean, I don't work in the US system, but I mean, you know, looking at the costs of treating CLL, £200,000 10 years ago, or dollars rather, $600,000 now. This is per patient. So the affordability of this in most healthcare systems is really questionable. And then tolerability. Is ibrutinib actually the right thing to be used? Because we know that ibrutinib fundamentally is a well-tolerated drug. In the context of 10th remission that was seen in the original data, having a drug that gives you a 2/3 chance of actually having a long-term remission is fabulous. And the toxicities almost don't matter because the alternative is progression likely to the point of death. In the context of frontline treatment, whereby you're taking somebody that's often often quite well that might be on it for an extraordinarily long period of time, toxicity is an issue. And this was some retrospective data that's been looked, a number of series, both from trials, from the Resonate trial, some single-centre data from big US centres, and real-world, if US academic centres are real-world, which I might question, but significant discontinuation rates, up to a third of patients on ibrutinib discontinued. Now, it's a mishmash, it's retrospective, it's not been controlled, but nonetheless, the major reason why people are discontinuing ibrutinib is not due to the fact that they run out of money, it's due to the fact of toxicities. So consequently, this really puts into focus, is long-term ibrutinib actually the right thing to do? So CLL trials, if we just look at what standard things have been approached, and I'll just pick up on some of these. The first is an artificial distinction, fit or non-fit. Slow go, 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 however you want to put it. Is that actually valid anymore? Because one thing that we've clearly got now is that we have the ability to give more patients, irrespective of comorbidities, effective treatment, whether that's chlorambucil obinutuzumab or increasingly using BTK inhibitors. I'm not really going to pick up on that, but that distinction is probably going away as we're starting to use better tolerated and more effective treatments. We've focused on p53 dysregulation as being an area of unmet need. It still is. That's something that we still haven't fixed that. But there are clearly more genetic factors that we might need to look at. Most studies are looking at progression-free survival. Most of the trials that we do don't make people live longer. They do not improve the overall survival from patients. Progression-free survival as a surrogate endpoint. It helps regulators, it comes in more quickly, it makes trials less expensive. Is it truly the right endpoint to be looking at it? And then should we just be treating in the frontline setting forever, or do we have to look at tolerance and what we're doing with the therapy? So just in terms of something, it's a bit complicated, but just to come through it, this is looking at the genetic landscape in CLL. So CLL is a genetic disease. When one asks patients, they get traumatized by that concept in terms of, have I inherited it? Am I going to pass it on to my children? Not usually. But CLL cells are genetically abnormal. That is why they are CLL cells and not normal lymphocytes. And most patients, I hope, will be having their carrier type done, looking for evidence of FISH abnormalities. So this is looking at trisomy 12, common abnormality, deletion of chromosome 17, p53, deletion of the long arm of chromosome 11. Standard things that have been done for a long period of time. And you will find recurrent abnormalities in a good proportion. Increasingly, however, we're looking at genetic profiling, personalising our treatment and looking for things such as immunoglobulin heavy chain status, mutated or unmutated, or looking at specific mutations in genes. This is just picking out three. Notch here, SF3B1 here, or P53, the actual mutation itself. And this is just a series of patients in a retrospective, and this was looking at correlations, and it's not just about P53 deletions. So you can see here in red, in this group, a proportion of patients, all the red ones, each line is a patient, had 17p deletions. But you can see that also there's a correlation with p53 mutations. A smaller proportion have 11q deletions, but a larger proportion of these will have SF3B1 mutations. So we need to look at the complexities of CLL and trying to work out how we target our treatments, because at the moment we know that p53 is a problem, but also that underestimates what we're doing. We just chuck a treatment in. Some do well, some do better. We can pick these out. These patients with mutations here will do worse. Can we target them? Another question. But nonetheless, I think we should be using this more intelligently rather than just a one-size-fits-all. And then what's the endpoint? Progression-free survival. Is that the best endpoint? Overall survival should where we're getting to. One reason we don't do trials with overall survival is that in the first-line setting, it takes potentially decades. The trials become too long, they become too expensive. What about minimal residual disease? Well, minimal residual disease, I would suggest, and I'd be happy to be criticised on this, it's pretty much axiomatic. MRD negativity with CLL is good for you. There is nothing to say that MRD, if you achieve MRD negativity, you will do badly. Ibrutinib, you rarely achieve MRD negativity. clearly it's an effective therapy, but if you achieve MRD negativity, it would be reasonable to believe you do better. And maybe combination treatment is the way to go. So I'm going to show something from a UK perspective. The Clarity trial, this was presented at ASH at the end of last year. This is looking at a combination of ibrutinib and venetoclax, starting with ibrutinib, adding in venetoclax, time-limited treatment, and this is looking at the proportion of patients who become MRD negative. If you look with ibrutinib monotherapy, very few patients would get there. In red, they're all MRD positive to start off with. As you go through treatment, month 8, month 14, month 26, more greens. They're starting to get MRD negative, and the dark green, the depth of MRD negativity is improving. So it is possible with ibrutinib-based therapy to get to MRD negativity. And with short follow-up, and this is in relapsed patients, this is not in the front-line setting, follow-up is short, only going to 2 years. 100% of the patients were still alive, and almost 100% of the patients were still in remission. So it's clearly a very effective strategy. So this is something looking at a UK perspective, coming back to, well, we've already proved it. Ibrutinib's better than bendamustine rituximab, or it's better than FCR as shown in the ECOG study. It's something that I think we're still looking at in the UK. So the FLAIR trial, which will be a very large trial, 1,500 patients, frontline setting, patients under 75, comparing ibrutinib venetoclax, the combination that we've looked at in the UK, or FCR, still not throwing it away because it still can be an effective treatment. It still uses PFS, but importantly, what we're building into this trial, built into it, is that for those people that achieve MRD negativity, they can stop treatment. So if you're MRD negative, then you can stop it, and this may be something that's working towards the issue of how we can rationally use the treatment without looking at it being on ibrutinib forever. I'm pleased to say it's recruiting very well and continues to recruit well. This is just the recruitment. The red line is what we project recruitment. The blue line is where we've actually got to, and we're aiming for this to be finished in the UK by the summer of next year. So it's something that's clearly feasible to be doing this, and it's something there's been a lot of buy-in to it within this country. And then what should we do beyond it? So should we just keep people on treatment forever? So this is what we're going to do when FLAIR stops. FLAIR will finish next year, and STATIC is the trial that we have planned. It's trying to address this question about if you're on ibrutinib in the front-line setting, do you take it forever or do you stop it? There are data, not particularly in CLL, but with other tumours, looking at the fact that if you just continue with therapy, you give a survival advantage to the cells that are resistant. Over time, you're getting rid of the cells that are sensitive to the treatment, but you are applying selection pressure so that more cells that are resistant will be growing through. And so this trial, which we'll be starting next year, is looking at patients on BTK inhibitors. They've been on it for more than 2 years and have achieved a clinical complete remission, and then asking the question about whether we continue forever, standard, or whether we stop, monitor for progression, and then restart at the point of progression. So looking at then coming back generally, trials are good. This is how we make progress. Why don't more people go into trials? We've already heard from a US perspective. I mean, the published statistics suggest that about 3% of cancer patients in the US are in trials. Why is that? I don't work in the US, so I can't tell you. This is just something that comes from what's in the literature. This was presented at ASCO a few years ago. So about 50% of the case, there isn't a trial. We need to have trials in order to make things better. In 20%, we looked at its clinical factors in terms of eligibility. We make the trials wrong. We make them too stringent, or we have issues whereby we're addressing the wrong issue. So we've got a good trial, but then someone decides that we're going to have the wrong inclusion criteria, so we can't recruit to it. In about 15%, it's me, it's my colleagues, it's physicians who decide, I know better, I'll give the right treatment. I've got ibrutinib available, so I'll just give it. And then 15% of it is patients. And I don't use this as a criticism of patients. There are valid reasons, but not everybody who is offered a trial will wish to go into the trial. Some of it comes down to cost, it comes down to their views on it. We all have the right to be able to look at things and say yes or no, but it's complicated. But how can we improve this? Well, sorry, this is just coming back to a US perspective. What about in other healthcare economies? This is looking at worldwide non-US. and looking at the rank of why investigators believed that they had issues with trial recruitment. This is not CLL, this is just cancer trials. The lower the score, the more of the issue. And the standout in terms of in less well-funded healthcare economies is funding. Trials costs money, so you don't have trials if you don't have money. So this is a sort of issue of the rest of the world. In the UK, in terms of what can be done, I'm pleased in the sense that we can make things better. It's sort of advert for the UK. The National Cancer Research Network was established in 2001. Prior to that, this is cancer trials, not just haematology, certainly not just CLL, 3.7% trial recruitment in cancer trials, about the same as we're seeing in the US. And by building infrastructure of actually connecting centres, putting them together, cancer trial recruitment has increased significantly within the UK. So it can be done, but it requires a coordinated effort between investigators and between centres. This is clearly something that working in the NHS makes it different than working in other healthcare systems, but it is possible. And where we're at in terms of haematology oncology trials, well, this is looking at where we get to with this. So about 20% of patients in interventional trials, about 40% in non-interventional trials. So we're doing pretty well. But before putting myself on a massive pedestal, cancer outcomes in the UK, we're right up there with Croatia. This is not CLL, it's not haematology, but this is looking at cancer survival. And one of the issues that we have in terms of making patients better is not addressed by trials, it's about presentation. So one of the issues we have in the UK is people present late. And consequently, all of this is sort of academic. In patients who are presenting with very advanced disease, it immediately makes it more difficult to treat. So something else that we need to be focusing on. So I'll leave it to you in terms of, if you want to know, what did I do with the patient? Well, you can suggest what I might have done. We do need to do the right trials, and I think it's extremely difficult at the moment because of the profusion of data and the number of new agents and the short follow-up. How do we put this together in terms of saying what's right? There are a number of barriers to recruitment, and they're different between different regions. Some of them is physicians, patients, the infrastructure, but it needs investment. And I think my view on the future, for first-line therapy, we need to be integrating our prognostic biomarkers into things because it's not a one-size-fits-all, and there are new agents coming along so we can use treatments more rationally. Drug combinations, I suspect, are going to be the way we're going to be going because of trying to achieve better depth of remission with time-limited treatment. And I'd suggest that we should be looking at MRD as an endpoint. Better depth of remission will lead to longevity. And if our aspiration for CLL is cure, we have to be eradicating the disease. We are never going to cure CLL with treatments that just put people into long-term partial remission. Thank you.